Hi, my name is Dana Pasquale. I am in the Duke Department of Population Health Sciences, and I'm going to talk to you today about molecular epidemiology, so recreating networks using epidemiological and phylogenetic information. So first I'll give an introduction to phylogenetics and pathogen gene sequence analysis, talk about applications of molecular epidemiology in public health, then talk about implementation and methodology if you are choosing to embark on this kind of work, and then give two examples, so HIV contact and transmission networks, and then a network of patients who have a hospital-acquired multi-drug resistant bacterium. So first, pathogen gene sequence analysis. I'm going to try to keep this as brief as painless as possible. So most of us have probably seen a tree of life image where different animals are represented in the evolutionary tree. So this is a phylogenetic tree, which is a visual representation of the amount of genetic similarity of different organisms, specifically African antelopes. So they all look pretty similar. We used to use morphological characteristics to classify organisms, but now we use genetic similarity. So how do we get to this? So recall that our genetic material is made up of A's, T's, C's, and G's. These are nucleotides. A set of these nucleotides all together in order as they were sampled from an organism is called a sequence. And if you can obtain sequences of these nucleotides that more or less start in the same place in the genome, then you can line them up so that there is the least penalty in the change in nucleotide from one sequence to the next, and assign a similarity based on genetic similarity. So that's called an alignment, and that is what is shown here. And the method of similarity chosen depends on your organism and your goals, and I'll talk about that soon. You can then look at the similarity of sequences to build a tree where the branches are based on divergence among the sequences using maximum likelihood or Bayesian processes. So using the alignment, you can create a phylogenetic tree, which is a visual representation of the amount of genetic similarity of different organisms, similar to the way that a tree of life diagram is a measure of morphological similarity. So each makes an inference about common ancestors, which are unsampled, and those are at the nodes. So the branch points, these nodes are the inferred common ancestor, and again, those are unsampled, and it's based on counting backwards, more or less, from the divergence from the two sampled organisms that you have. So in these diagrams, it's the amount of lateral distance that tells you the amount of genetic divergence. The vertical distance is meaningless and can be changed. It can be stretched or shortened or even flipped around just to improve visualization. In any tree, you want an appropriately chosen outgroup, and that allows you to see how clustered your group of interest is. So the outgroup should be distantly related enough that you can see similarities, but not so distantly related that everything looks like one big cluster relative to the outgroup. So we can also do this with infectious pathogen gene sequences collected from different people. So the slide that I showed, this slide is actually made up of publicly available HIV gene sequences sampled from different HIV positive people. And the gene sequences are aligned so that they can be used to group people together by sequence similarity. And then a measure of genetic divergence, which can be a percentage or an absolute, and can be weighted differently across different parts of the genome, is calculated to determine similarity between the infections in any two people. And this similarity is used to develop inferred transmission clusters, which are groups of people with like HIV sequences who are estimated to be close together in the transmission chain. So this phylogenetic tree is a visual representation of the likeness of HIV gene sequences collected from different people. So each terminal branch, each point at the outside of the circle, is an HIV sequence, and each sequence was collected from a different person. And the sequences here are used to group people together by sequence similarity to develop inferred transmission clusters, which are groups of people with like HIV sequences who, again, are estimated to be close together in the transmission chain. And so the idea is that since the virus mutates over time and as a result of selective pressure from people's immune systems, looking at change in virus between people can help you get a sense of spread through a population over time. So in this tree, samples were collected in North Carolina, 1 per person from 1997 to 2014. There are several key limitations to these kinds of analyses, though. So first is that you can't infer direct first-degree partnerships. There could always be an unsampled person intervening any 2 people who are in a cluster together. And there are 4 clusters picked out here, and those are grouped by color. There's a red, a blue, a purple, and a green. So since we can have unsampled people, we can't say that any 2 people in a cluster together, even if they have very similar infections, were actually partners with each other or were together from one person to another in a transmission chain. We also can't infer directionality of transmission. We can't say that person A transmitted to person B, for instance, in this kind of analysis. And of course, a limitation is having incomplete sequence coverage because it limits the inferences that you can draw, again, because there could always be unsampled people. There's also no clear consensus on a relative percent difference to use, and I'll talk about that in more detail later. It depends a lot on the data that you have, the conditions of your study, and the research question that you're asking. So on its own, this kind of tree isn't very informative. It helps to have epidemiological or contact data, which can be used to create a network combined with these data. So, and having network or epidemiological information helps with some of the limitations. So we can know, for instance, who might have been partners, else you always have to worry about the intervening people who weren't sampled. And then the directionality of transmission may still be an issue, though. With enough people, though, the epidemiological or network information, especially over time, can help sort out where transmission is happening and get ahead of it. And that's the main goal of contact tracing. So the field of molecular epidemiology aims to make use of these types of data. So briefly, in order to best apply pathogen genetic data to understand disease transmission networks, and that's specifically networks or clusters of people with like infection to understand person-to-person transmission with some amount of granularity, the mutation rate of the pathogen needs to be in a sweet spot where it doesn't mutate so quickly that we can't keep up with it, like some influenza, or mutates so slowly that it isn't informative, as in the case of the bacterium which causes syphilis. At least one region of the genome needs to either be responding to selective pressure as it's transmitted or have a high copy rate and enough mutations that it differs as it moves from person to person or differs over time. So HIV fits the bill here, so does Hep C virus and a couple of other viruses. So HIV is a single-stranded RNA virus. It copies fast and it's very sloppy and it mutates a lot. There aren't any checks as it replicates, and some of the mutations happen to be viable. And I'll talk about this in a little bit more detail going forward. So to understand transmission networks, which is one of the ways that molecular epidemiology is used in public health, it's commonly used to understand transmission networks, to confirm outbreaks, or to guide interventions. So understanding transmission networks, I think probably a lot of us over the last 2 years have seen these kinds of graphs for SARS-CoV-2, the COVID-19 pathogen. So this kind of analysis was also used to better understand the 2014 Ebola outbreak in Sierra Leone. And then also I'll show a paper looking at an outbreak in a burn unit. So this is from NextStrain, which is a website that collects sequences for SARS-CoV-2 and a couple of other pathogens. And this was from March 30th, 2020. And at the time, there were just over 740,000 cases worldwide. And what they were doing was looking at the change in genome by location to try to estimate how much SARS-CoV-2 was moving around the world. And then, a little bit more than 2 years later, on April 16th, the next strain is still going at it, where instead of being at 748,000 known or presumptive or confirmed cases worldwide, where we were at more than 504 million. But if you look at the circled red, this entire graph is only made up of 3,000 sequences. So, one of the limitations of this kind of analysis is the amount of data that it takes to do it, and then not having, most people with COVID don't have a sequence done. But still, using just this very limited information, we can look, the pie charts here are by variant. We can look at how much similarity there was between variants across regions of the world. So with the right data, though, and under the right conditions, instead of looking at the broad strokes of movement of a pathogen over time, you can really drill down and try to recreate or understand transmission networks. So the paper here, the first author, Kanamori, I highly recommend this paper. They use molecular epidemiology to understand transmission of Acinetobacter within a hospital burn unit. So it's a hospital-acquired infection among especially vulnerable people, and it was over a span of years. So the nodes here are colored by the date of infection, and it spans October 2007 to July 2010. And they were noticing that the patients were becoming infected with Acinetobacter and were trying to figure out the source. So they used the sequence of the pathogen. They used detailed information about the locations and care teams of the patients who were in the hospital, and they're generally there for a length of time. And then also environmental sampling. And they combined the molecular and epidemiological information to create a network, and then refined it using Bayesian methods to estimate a final putative transmission network. So what's interesting about this is that the Acinetobacter analysis identified three sequential outbreaks in the hospital instead of a single long-term outbreak. And these are the three the three reconstructed outbreak chains identified are displayed. So again, the nodes are colored by time. The first one starts with the big red node A03. And then toward the ends of the component on the top, there's an orange node in the bottom right. It's called A15. And so that is actually the start of a second outbreak. And it was considered a second outbreak because the patients were infected with a bacterium that was the product of recombination between the bacterium responsible for the first outbreak and another one that was also co-circulating in the hospital at the time. And then an analysis of patient co-presence and movement through wards also confirmed that the second outbreak was due to environmental contamination. And then the third outbreak was ongoing for a while, and that's the component at the bottom starting with A20. And it was a completely different subtype of Acinetobacter. And this is really an excellent paper. It's very well done, and the figures are very illustrative. So I do highly recommend this paper. So phylogenetics can also be used to confirm an outbreak. So since the SARS-CoV-2 pandemic, we're probably all familiar with the idea of contact tracing. So it's a strategy for infection containment that involves public health personnel interviewing the case and then figuring out how to locate and test all of the people who might be at an elevated risk of being infected based on contact with the case. So for HIV and syphilis, that includes generally sexual contacts, though HIV might also be needle sharing contacts. And then, there are some other rarer ways to transmit HIV or syphilis. So a physician at UNC, Ann Dennis, and I were asked to assist the NC Department of Health and Human Services because they observed a surge in acute HIV in the western part of North Carolina. So people with acute HIV, they're early in their infection and they're highly infectious to other people because the immune system hasn't responded yet to tamp down the amount of virus. So if they do have a sexual relationship with someone else, then they're very likely to transmit to that person. So NCDHHS was concerned that there might have been an outbreak occurring as a result of rapid serial transmission from one highly infectious, acutely infected person to another, just onward. But they weren't sure if it was definitely an outbreak of acute HIV versus an increase in diagnoses during the acute phase, which is actually an indicator of better case finding and is a success. So Anne and I collaborated to compare the contact tracing or epidemiological links between cases and their contacts with their HIV transmission clusters. And since people with acute HIV infection were only recently infected, they're more likely to cluster in a phylogenetic analysis because their infections haven't diverged yet through lots of mutations accumulated over time. So this is a multiplex network, which we created in response to NCDHHS's request to assist them in the outbreak investigation. The solid black lines depict partnerships disclosed during contact tracing interviews, and that's the epidemiological information. And then the dashed green lines depict having very similar HIV gene sequences, which indicates being very close together, though not necessarily first-degree partners in the HIV transmission chain. So the green lines are the molecular information. And then, as I said, NCDHHS was wondering whether the rise in diagnoses of acute HIV infection was serial acute transmissions, rapidly onward from very infectious people, which is bad, or a better case finding where people are diagnosed closer to the time of infection, which is good. And then, as it turns out, we were able to show with this diagram that it was the latter. So better case finding closer to the time of diagnosis, which is difficult. And so that's great. It's a success. Had there been serial transmissions, then the components would have been in the same transmission cluster and linked together by the dotted green lines. Instead, the majority of the components are small and discrete and largely not phylogenetically similar. So this was an example of when molecular epidemiology was used to confirm that there was not an outbreak. So a molecular epidemiology is a promising area for guiding interventions. So Anne, the physician at UNC, is using near real-time analysis of phylogenetic clusters in North Carolina to determine where contact tracing, which is a containment strategy, should be focused, and that's the HIV prompt study. And then there are a few hospital consortiums which are tracking multi-drug resistant organisms among inpatients so that control measures can be implemented when necessary. And the first one listed there, Crackle1, I'll talk about at the end of this presentation. So I'm going to talk now about how one does these types of analyses. So the applications are broad, and it's a relatively new area of research, so applications are still being developed, especially as computing gets faster, particularly on big data. But of course, the data are frequently the limiting factor. These are all typical study design issues, so the sampling frame, whether the population is appropriate, but in this case, it's both the host population and the pathogen that you need to think about. So is the pathogen appropriate is an important question. So I said that there's a sweet spot for mutations, and some pathogens hardly evolve and thus aren't good candidates for recreating a transmission chain because you don't see enough differentiation between hosts. In some cases, you may be able to adjust your goal to find clusters instead of recreating a transmission chain. For instance, that's the kind of thing that's done when we're looking at a foodborne outbreak like E. coli or Salmonella. It doesn't matter really who gave it to who. Knowing that everyone has the same bug or the same pathogen is what's important, especially if you can identify the same bug at a source, at a lettuce processing plant or something like that. And then some pathogens mutate so quickly that you really can't match from one person to another. And it doesn't end there. So the choice of gene within the pathogen also needs to be considered. So the reason that pathogens mutate isn't because of anything that they're trying to do. They are copying, they're replicating themselves, and then there are mistakes during the copying phase of replication. Some of those mutations aren't caught, they happen to be viable, and then some of those mutations happen to confer an evolutionary advantage under the conditions at that moment. So for instance, some mutations are actually costly in terms of energy or replication to a pathogen, But if they evade certain antibiotics or antivirals, as it may be, and the person is taking those drugs at that time, then the ones which evade the antimicrobial treatment are selected for. So under the selective pressure of the immune system or treatments, certain mutations stick and then get passed on when that version replicates. So the gene selected needs to account for those changes. So this is the HIV genome. HIV, as I said earlier, replicates very quickly and it's sloppy. There are no checks in its replication. So inevitably there are tons of mutations and you know, they work sometimes. They're an advantage or they're at least viable. And so at the very least, if there are viable mutations, we can see changes between people. So the orange gene is polymerase or POL. It's highly variable because it's the target for drugs. Selective pressure because current guidance is to treat every person with HIV as soon as they're diagnosed, because there are significant improvements in health, plus it drastically reduces the likelihood of onward transmission. Clinicians in countries with sufficient resources generally have the newly diagnosed person's POL gene sequenced to check for resistance before prescribing a drug regimen, since drug resistance is encoded in that gene, and this is such a highly studied pathogen that it's well understood. So that means that POL is therefore a very convenient choice because it's more available than other parts of the genome because it's sequenced per clinical care. And then it's appropriately variable that transmission clusters can be constructed. Another commonly used gene is N for envelope because it's the target for the immune system, so there's selective pressure there. To bring this home a bit more to our present world, this is the SARS-CoV-2 genome. The green part of the genome on the right includes spike, that's S, which is the mRNA vaccine target. And then it also has nucleocapsid, that's N. So both spike and nucleocapsid are immune system targets. So both have selective pressure exerted there. And then this is a network that I'm working on right now. It's a bacterium called Klebsiella. And these bacteria have a core genome, but they also have the ability to pick up or drop accessory genes, extra genes that encode for resistance. They're not necessary for life. So the core genome doesn't change very much among people who have this infection. And so if you were to construct a network of people with Klebsiella looking at the core genome, it looks like this. So everyone with, where everyone with a similar bug, and these are very, very closely related, are tied together. And so you end up with these clumps like this that are very, very dense. So the color of the edge is by time of diagnosis. The node color is just the facility where the person was diagnosed. So you can see that many of the people in the cluster, in the component full of red nodes, they were all in the facility at the same time. They were all diagnosed around the same time, which you can see by the edge color. And they all have very similar infections. So the likelihood of determining a transmission chain is low, given the lack of pathogen evolution in the core genome. And so adding epidemiological information to this kind of network is really helpful. And so back to here, just like a network, there needs to be sufficient coverage in one area of time and or space for connections to be observed such that you get a sense of the big picture. So since the pathogens evolve, the coverage by time or space is important, else you lose potential edges if they're defined by the pathogen. So some pathogens are better studied than others, in which case the mutation rate across different parts of the genome may be known. It's usually modeled as a constant rate. And then the inference can be drawn across longer time periods. So, and now something important to think about is making sure that you consult your statistician. Of course, they always want you to involve them at the time of the proposal, not once the data are already collected. But then in these types of analyses, you also may want to add a bioinformatician or a phylogenetics methodologist to the early consult set. So also make sure that that person is versed in your organism of interest, because a rapidly mutating virus is very different than understanding a more complex bacterium as it mutates and replicates over time as well. So we can talk about how to define the edges in a phylogenetic network. So first, the relative contribution from the epidemiological or contact data versus the molecular information is important. It depends on your research question. And then I'll show some work at the end where the molecular information is treated as the gold standard because we know that the epidemiological information is poorly observed, and that's in HIV. And so even where we don't have epidemiological information of contact between people in different contact network components, the phylogenetic information, when you construct a cluster, can tell you where you're missing information in the contact network. But then for something like Klebsiella that I just showed, we definitely want to use a healthy dose of epidemiological information because the molecular information is more informative to say a link didn't happen than to say that a link did happen, given the slow rate of change in the core genome. So it's better in that sense for the epidemiological information to take the lead and then the molecular information to confirm that we didn't miss something. And then, of course, there is a need to be really careful about the limitations of both types of data, especially as both tend to suffer from the same issues, namely poor observation of either party involved means that an entire edge is missed. And then from here, the edge definition is going to be limited by the data that you have and then what you can define afterward. So genetic distance can be an absolute number of changes or a percent difference. It's usually basically divergence quantified. And then it can be pairwise from one sequence to every other sequence in the data set, or each sequence in the data set against a model sequence that you're comparing against. Another important point is that the genetic distance cutoff is a function of what can be observed based on the study design. So these pathogens are subject to selective pressure from the environment, which could be other microbes if they're free living, else it's the host immune system, or even treatments from pharmaceuticals. So the divergence is evolution over time or per transmission. So to go back to looking at the research question itself, often the pathogen, the population, and the ability to observe will dictate whether transmission clusters, which are two or more people with like infection, versus chains, which is looking at a putative, an estimated movement from one person to another of the pathogen. So the research question, the pathogen information, and your population will determine whether you can look at clusters or chains, and then you design your study based on what you want to know about those things. So recall from earlier that most work with HIV uses clusters, but that's a function of the data collection process in most cases, which is often at a population level and has incomplete coverage, thus directionality of transmission, and the potential for having an unsampled intervening person is always there, especially when you're looking at everything retrospectively. You're taking people who are already diagnosed and then you're using their clinical care sequences. It's not a prospective or active data collection process or an active surveillance process. However, this 2011 paper was really important in the field of HIV. The investigators followed 1,763 couples where one person had HIV and the other didn't. So they knew at the beginning who was positive and who was negative. And they treated half of the couples, they treated the HIV positive person with anti-HIV drugs immediately after diagnosis, and then followed what was the standard of care at the time for the other half of the couples, which at that time was to only give anti-HIV drugs when certain signs of HIV progression were observed in the infected person. So they followed these couples over time. They tested the negative partner frequently and were able to identify a small time window if any transmission to the HIV negative person in the couple occurred. Then they confirmed with phylogenetics whether the infection was linked to the HIV positive partner in the couple, or if the HIV negative person in the couple had been infected by someone who wasn't their main partner. Because the couples were observed regularly and they knew the HIV status of this person at multiple time points, they used a really small genetic distance cutoff. It was 1.5% genetic distance to say that a transmission occurred between the HIV positive person and then the newly infected person if a transmission occurred in the couple. And that could be done because they were observing these couples in near real time, and they would know that the pathogen hadn't had much time to mutate away from the donor strain. So again, I'm going to make a plug, but that's why you talk to the bioinformaticians early. So choosing a research question, you have to think about whether you're retrospectively assessing transmission, if you're aiming to create a network, if you're estimating evolution and travel from one place to another of a pathogen or organism of interest. And then those things will guide the choice of genetic distance cutoff within the parameters of the organism or the pathogen and gene, and within the limitations of observation. So as in any research project, you've got your population, and then as in any network research project, you've got a definition of relationships between people. The data can be really expensive to collect as there are sampling and sequencing costs. So there are resources where the data are already available. So two of which are listed here. They're repositories for genetic information. So funded researchers agree to deposit some or all of what is sequenced with that funding from their studies into these databases, which are then made publicly available. There are a lot of sequences in these databases, but you get what you get. And there is limited information about the source of the sequences attached to the sequence, but they're worth looking at. So then just like any other network, you've got your nodes, which are defined a priori as is your edge definition. And then edges can be weighted or unweighted. So here's the Klebsiella network again, but here the edges are weighted by a transmission probability that's a function of not only absolute genetic distance, but also facility co-presence and the amount of overlap between any two patients who shared a facility. So the data set for this is an N by N matrix with weighted edges, which have a transmission probability. So additionally here, I'm showing there are some people who are connected by dotted lines, and those are people who had very small genetic distance, but were never observed to be co-present at the same facility. So as in any other kind of network, you can also have multiple edge definitions. So now I'll show a few examples, starting with HIV in North Carolina. So North Carolina is fairly typical of the US South when it comes to HIV. So there are about 60 new cases per 100,000 adults or adolescents per year. There are a lot of disparities in the new case rate. So it's highest among people aged 20 to 29. It's highest among Black or African American people, and it's higher among men who have sex with men. And then when you look at the new case rates, there's fully 5 times higher among Black MSM than among the state as a whole. Most transmission is sexual, but like a lot of other places, we are seeing an increase in injecting drug use due to the opioid epidemic. And so that could definitely change soon. So previous sexual network studies of North Carolina found that a lot of people who were diagnosed with acute HIV infection have partners that had a known diagnosis previously, but a lot of the partners weren't in care. And if you do take HIV drugs and you're on the right drugs and you're adherent to the regimen, then you're really, really unlikely to transmit sexually to your partners. So we wanted to look at where we might be able to intervene in the network. So a study that was done outside of North Carolina was looking at the viral load, which is the amount of virus in any one person kind of averaged over the entire network And it's an indicator of the number of people with uncontrolled HIV, and unsurprisingly found a correlation between network viral load and then the risk for an HIV negative person acquiring HIV. So we went back and we took some of the contact tracing data that were collected during public health investigations, and just like COVID, the goal is to try to get ahead of the infection and get everyone tested and treated, and then, to try to prevent more onward transmission. So in North Carolina, this is all done manually. It's not done with, apps or tracking. Instead, someone called a disease intervention specialist contacts the person who's newly diagnosed, interviews them to try to ask about, any kind of care needs that they might have, also asks about risk behaviors, and then any kind of contacts or partners who might benefit from testing. So, so these are called partner notification interviews. It is a process that is very similar to discriminative snowball sampling, because you stop once you find people who are HIV negative. You generally don't ask them about their partners unless they seem to be in a really high risk set. And the network edges are right there with the HIV negative people. As long as you keep finding people who are HIV positive, then you'll keep interviewing, especially new positives, if any are diagnosed during the contact tracing process. So missing data, though, is the biggest threat to the validity of these kinds of analyses. So around a quarter of people in any given data set will refuse to be interviewed. We have a lot of partners who can't be located. And then even if you do find someone who's newly diagnosed with HIV, a lot of the times you can't find a contact who was HIV positive. So we know for sure that we're missing some of the partnerships there. So assuming you have this kind of network at the bottom, you also, one thing that you do is that if someone is previously diagnosed, you don't actually interview them again. You only interview them at the time of their diagnosis. So we know that when we are interviewing people, sometimes people could have been infected a really long time ago. And so the partnerships might not reflect the time of infection. And so we might see something that actually looks like this, where we're not observing the gray nodes because the person wasn't interviewed or the partnerships that of interest occurred too long ago. So instead, we wanted to know where the gaps were in the contact tracing network. So where we were missing people, since we were seeing a substantial number of people who were newly diagnosed who didn't have any HIV positive partners identified. So we used the HIV transmission cluster information as a gold standard for the network. So, so we did that because we knew that any HIV cluster information that we had wouldn't have been limited by people's choices to talk about partners, and it wouldn't have been limited by the case definition, which is usual for someone who's newly diagnosed with HIV, which is that you don't ask about partnerships that occurred more than a year ago, despite knowing that a fair number of people were infected more than a year ago. So the network here are people who were in the sociosexual network, and some of them are social contacts who the person said would benefit from testing, which is why we call it a sociosexual network, among those investigated by the disease intervention specialists. Here, our transmission cluster definition was less than 3.5% pairwise genetic distance across all cluster members. And recall that I said that the study where they prospectively followed the pairs, the serodiscordant couples where one person was HIV positive and one person was HIV negative, was a 1.5% genetic distance because of the way that the study was designed and the amount of observations they had for each of these couples. Instead, once you're using the contact tracing and the clinical care information to reconstruct the transmission clusters, some amount of time has passed. We don't know that we have actual partners in the data set. And so we used a 3.5% pairwise genetic distance. And this was set against a background of 15,000 whole gene sequences that were collected from unique patients across North Carolina. It's the same data set as that circular phylogenetic tree that I showed at the beginning. So this network was among people who were diagnosed in the green counties. There's 11 green counties that make up HIV control region 6 in North Carolina. So they're contiguous counties. They include both urban, rural, and peri-urban counties, and they're fairly representative of this region as a whole. So what we were doing was we were saying that, for instance, if we had this phylogenetic cluster where people A, B, and C are all fairly close together, they're in the same transmission cluster, this is where they are in the tree. But then if we were looking at the epidemiological contact tracing information, and we learned from interviews that A and B were connected to each other, but C was not connected to either of those people despite being in the same transmission cluster, the options for what we missed were that either C is a partner of A, and we missed that. C is a partner to B, and we didn't observe that. Or there was an unsampled person who was in turn connected to both C and either A or B. So this is the sociosexual network of people that were diagnosed over 2 years in those 11 counties in central North Carolina. So that included 280 index cases and 383 of their partners. So that's the N equals 663 at the bottom. And the edges are based entirely on epidemiological information, which in this case is partners disclosed during contact tracing interviews. So the circles are men, the squares are women, and the triangles are trans women. So the node colors here represent the transmission cluster in which the person was included after independently constructing the phylogenetic tree and then mapping the network members onto that tree. Or if they weren't identified as part of a cluster, then white through kind of the brownish color say something about the person's HIV status. So we wanted to look at the clusters that were, whether the clusters were completely contained within one network component, or if they spanned multiple network components like this red cluster I that I've picked out here. Because perfect partner elicitation means that people who are in the same transmission cluster should at least be in the same network component. So looking at a few of those clusters, including red cluster I a little closer up, here the sociosexual network connections are represented by solid lines, and then the dashed lines represent 1.5%. So this is the same definition used by the serodiscordant pair study. So it's 1.5% or less pairwise genetic distance, which is pretty close. It's closer even than our cluster definition. And those are the dashed lines. So there are some nodes here that have X's in them, and those were people who were in the transmission cluster but didn't meet the definition of being in our sociosexual network because they were either in a different administrative area, a different region in space, or they were diagnosed at the wrong time and not elicited during the partner tracing interviews. So to walk through this, looking at the red cluster I in the top left, there are 4 network members in cluster I across 2 components, and those are the people who are red circles that don't have X's in them. But there were 8 people in the cluster statewide because there are 4 more red nodes who have X's in them. So had partner notification completely observed the network, there should have been some direct or indirect link that connected both components with network members into a single component. Instead, the component with 1 red cluster member isn't joined to the larger component despite being within 1.5% genetic distance of all of the other red nodes. So doing a little bit more analysis, there were 446 contact tracing relationships in the network. So most of them involved 2 males or a male and a trans woman. Most involved people of the same race, and the majority of edges included at least 1 Black person. There was no difference in age or gender with respect to having a sequence for analysis, but white people were more likely to have a sequence than people of any other race, despite being a minority in the data set. I think that the most important finding here is the last one, because it indicates that most of the male-male partnerships observed weren't in the same transmission chain, despite both being HIV positive. So along the way, we have men who are being infected by people that we're not observing during contact tracing. So the infection could have happened a while ago, and the partnership was before the period of interest, or there's mistrust and people aren't disclosing, or we're just not asking the right questions. And then the racial disparities in having a sequence is also telling. So having a sequence is a marker for being in HIV care, because the provider orders the lab for you at the first HIV clinical care visit. So in a lot of cases, it means that people aren't even making it to their first HIV care visit. So a second project where Jim and I this project, Jim and I are looking at multi-drug resistant organisms acquired in healthcare facilities. So some multi-drug resistant organisms are already a health crisis in the US, and that includes carbapenem resistant Enterobacterales, or CRE. They're resistant to first and second line treatments. And so since there are limited treatment options, there are frequently poor outcomes. Most CRE infections in the US are due to carbapenem resistant Klebsiella. And then Klebsiella, I said that it has a core genome that doesn't change. And so the plasmids are some of those accessory genes. And a lot of Klebsiella that is sequenced, you can see that they carry up to 3 distinct plasmids, though in some cases up to 10 have been observed. And then CRE burden is correlated with facility to facility connectedness, and I'll show that in a minute. So the CRACL-1 study is the Consortium on Resistance Against Carbapenems in Klebsiella and Other Enterobacteriaceae. It was a prospective multi-center study. It included 966 patients with CRE who were enrolled from 18 Midwestern acute care hospitals over a 4.5-year period. So the PI is David Van Dyne at UNC, and along with the sequence, they collected admission and discharge dates, testing dates, locations where the patient was before admission and upon discharge, the body site of infection, strain test typing results, and then susceptibility to drug test results. So starting with the 18 acute care hospitals, this is what the facility connectivity looks like. So the blue nodes are the acute care hospitals. Each dot or node is a facility, and they're colored by facility type. The edges or connectors indicate a patient transfer between facilities where that patient was diagnosed in the acute care hospital. And the width represents the number of patients shuttled between the facilities in either direction. And that's because a lot of the patients were in some kind of residential care facility, either before admission, post-discharge, or both. So edge width is color shifted from black to red based on the proportion of people who may have been infected when they were transferred based on their test results during their admission. So this is the network of putative transmission events between patients based on facility co-presence and Klebsiella the difference between their Klebsiella infections, their genetic information. So here the nodes are people, and transmissions were inferred based on facility co-presence and similarity of sequence. So the edges are colored by estimated likelihood of transmission, where red is the most likely and black is the least likely. And then the CRACL patients that had no estimated transmission linkages aren't shown at all in this graph. And here they're shown by the location of the hospital, the state of the hospital in which they were diagnosed. So the next steps for the Klebsiella project are to look at sequences, including the accessory genes, to see if we can see a certain amount of divergence among those steps. So we looked at the transmission chains and we picked out the people who had a high degree of overlap, and we selected a few for the long read sequencing, so we can also sequence the accessory genes and see if we can get a better sense of transmission that way. So we're thinking about this kind of like a game of telephone, from the blue node on the left all the way to the last blue node on the right. We're going to see along this putative transmission chain if we can see a difference in the long read sequences in the accessory genes. So with that, I would like to acknowledge the sources of the data, the collaborators, and the funding for the projects described here. I'm incredibly grateful to all of these people and organizations, and that concludes my lecture. And for those of you who will be there next week, I look forward to talking to you about some of this or answering any questions that you might have.